Yeah, I think it's interesting that for most treatments that we use, there have been sort of dose finding studies to try to identify, what is an appropriate regimen to use, and that's a typical part of development and early stage research, and that has never been done with food immunotherapy, and so you have lots of studies that have just sort of arbitrarily picked different doses, and people that are practicing this in the community now sort of arbitrarily pick different doses. And so we actually haven't really worked out what an optimal regimen looks like. The regimen that was tested in the phase 3 study for AR101 will likely go forward, but even that was developed based on preliminary studies that were themselves quite arbitrary. And so I'm really interested in this concept. There was a recent study that came out from Germany using a product other than AR101, but with a target maintenance dose that was about a third as big, and with a much slower dosing process, showed that the safety of sort of a low dose, slow approach was very different than what is seen in other studies. In other words, much better. Fewer systemic allergic reactions, no eosinophilic esophagitis, very low rates of dropout. Just looks very different than other OIT studies. And conversely, we have data from people treating patients in the real world with very high doses of OIT that shows that you get toxicity. You have people who are well into their maintenance regimens having anaphylaxis and using epinephrine because they're taking 2 grams or more per day, which is almost like a little food challenge every day, and you have people reacting. So we have these quite wide range of doses that are being either tested or used, varying by orders of magnitude, and it's all empiric. We don't know what the right regimen is, and I think that's something that really needs to be worked out rigorously.